We lead the Annex 4 project for the U.S. Department of Energy and its international partners. So what I would like to do is just talk very, very briefly about what we're doing here today, and then ask people to introduce themselves. This is a forum looking at potential risks to larvae and plankton from tidal turbines. We deem this an expert forum as opposed to an open webinar because we have invited people to participate who have some thoughts, some input, some research relevant to this question. Annex 4 is an international collaboration under the Ocean Energy Systems collaboration of 23 nations. We have 13 nations presently participating, trying to understand environmental effects of marine renewable energy devices, that is largely tidal and wave devices, on the marine environment and its organisms and habitats. And what we are trying to do here today is really inform this question that comes up around tidal turbines. Is there a risk to larvae and plankton? And if so, what does that risk consist of? So what we're going to do, as you can see on this slide, we'll do some brief introductions just to understand who's on the phone. And then the rest of you will be muted while there's some presentations just to avoid background noise. And then we'll have a discussion at the end. Go ahead and type questions into the IM box down at the bottom of your screen, assuming you're on the app online, and we'll be able to get to those, but we will, open the mics again so everyone can participate. Okay, so what I'd like to do is just go through a very quick rundown of everybody introducing themselves, where you're from. We really don't need more than that. So again, I'm Andrea Copping with Pacific Northwest National Laboratory. So why don't we take it with our speakers first?
I'm Rayanne Miller, and I'm a benthic ecologist at the Scottish Association for Marine Science.
Hi, I'm Brian Sanderson. I'm a research associate with the Acadia Centre for Estuarine Research.
David Schlesinger. I'm a research associate at UMass Dartmouth School of Marine Science in New Bedford, Massachusetts.
Hi, I'm Brady Quinn. I'm a graduate student at the University of New Brunswick in St. John, New Brunswick, Canada. I study dispersal and drift of American lobster larvae.
I'm Anna Redden. I'm from Acadia University. I'm sitting here with Brian Sanderson.
Hi, Garrett Staines from PNNL in Sequim.
I'm Ginevra Hake-Klimash, also from PNNL, and I'm looking at environmental measurements.
This is Mark Bevelheimer from Oak Ridge National Laboratory.
This is Marshall Richmond from Pacific Northwest National Laboratory.
Mike Womble with Fisheries and Oceans Canada in Dartmouth, Nova Scotia.
Melissa Oldreeve, and I've got a few of us here from Fundy Ocean Research Center for Energy.
This is Nikki Sather from Pacific Northwest National Laboratory.
Samantha Eves from the Department of Energy, U.S. Department of Energy.
And I think that's everybody. Okay, thank you very much, just to give you a sense of sort of who's on the line. Okay, could you go to the next slide, please? So we have several presentations here today. They're going to be fairly brief, but to really try to get us all sort of in the idea of what we're talking about here. And in the order they introduce themselves, Rayanne Miller with SAMS will be talking to some background on the topic. What are these potential risks? Brian, who's a physical oceanographer, will be talking about the physics of turbines and pressure effects, and also thinking about this issue in terms of scale. Brady's going to talk a little bit about potential risk to lobster larvae specific to the Bay of Fundy, where, of course, you all are aware that there are tidal turbines slated to go in at the Force Test Center very shortly. And David's going to bring up the last presentation, talking about some studies they did of turbine effects on zooplankton. Some of the themes that we're going to try to think about through this forum, through the presentations, particularly in discussion afterwards, and then following up as well, is what do we know about the risk to larvae, both lobster larvae, but also fish larvae and zooplankton from tidal turbines. And think about, at the moment, most of us are thinking about single tidal turbines, but what happens when you start to scale up from single turbines to larger developments? Does this risk change? what level of uncertainty do we think we have associated with this risk? And what can we do to reduce that uncertainty? How can we address this risk? And finally, what research and monitoring needs to be done in order to really address this risk and determine what we really think about this issue? Okay. At this point, I want to turn it over to Rayanne, and go ahead.
Great. Thanks very much, Andrea. Thanks for, I guess, letting me give a bit of background here. What I'm going to do is just talk a little bit about the challenge, and then mainly to kind of kick us off, just put a few ideas out there on the table about some potential effects where there's been a few different pieces of work that have been done, and then some thoughts around kind of what the challenges are and where the uncertainty really lies. So I'm not sure what everyone's backgrounds are here, but I thought I'd start by mentioning the obvious, that working with zooplankton is something entirely different than working with megafauna. So the scales we're working on are entirely different. We're talking about millimeters is the scale at which these organisms in their larval stage experience the world, as opposed to feet and meters in the case of larger megafauna. If we're talking about hydrodynamics, we're now working potentially at low Reynolds numbers, where the viscosity of the water is completely different versus high Reynolds numbers for larger animals. So, you know, any hydrodynamic modeling collision risk, we need to assess that completely differently. We're also talking about organisms which might be mobile on a small scale, but not necessarily on a larger scale. So they are subject to the ebb and flow of water and currents. And I guess the other thing that I wanted to highlight is just the numbers as well. Talking about hundreds of thousands of larvae, eggs, zooplankton that might be in a body of water, particularly for those that are larval life stages. Assessing the population impacts of something like a tidal turbine, or any effects from a tidal turbine, is much more challenging. It's influenced by a number of different factors, and the population dynamics really aren't quite as straightforward, I suppose, in my opinion, perhaps. So I guess one of the key things is to think about generally the challenge that is scale. So I think potentially this is something that Brian might touch on, but the first thing that I wanted to bring up is the effects of turbulence. So there's been a couple of different studies that have talked about damage to zooplankton from changes in shear stress resulting from turbulence. So not necessarily in association with tidal turbines, but turbulence in general. And I think there's a lot that we can learn from some of these different studies. One of the ones that I wanted to point out, you see a figure from on the bottom right. And this was done by Mark Jessup, who's based in Ireland at the moment. But what you see is, so what he did essentially is he looked at zooplankton communities before a set of tidal rapids in the sea, and he looked at the makeup of that zooplankton community after the tidal rapids in the sea. And specifically, he was looking at mortality, so the number of dead zooplanktons before the rapids and afterwards. And he found a significant difference in mortality rates, or in the number of zooplankton that he found to be dead after the rapids versus before. But the key thing there was that it differed quite a lot between species, and for some species the difference was very little, and for others, so for example, you can see the third one from the left, Littorina littorea, there's actually quite a large difference there. So I guess one thing to think about is, how does this effect differ between the different types of species we might be considering, or how does it differ between larger zooplankton, so things like fish larvae, and smaller ones, so things like barnacles and other mollusks. And just another key small point is thinking beyond the larvae in the water column. When I guess if it's mesozooplankton, when they come to settle, settlement success in some species is also related to turbulence levels. So just another aspect of the changes in hydrodynamic turbulence from turbines to think about. And is there a potential there for changes in settlement success? I'm not sure, but something to think about. Another effect that I hadn't really thought about is the effect of turbine noise. Turbine noise is something that's generally very much associated with marine mammals, with birds, with fish, larger organisms. But I came across this quite interesting piece of work that was actually looking at the time to metamorphosis of crabs and how that was affected by noise. So what we do know is that for a number of invertebrate species, their settlement cues are guided by noise, as the same with a number of larval fish. So potentially, this is something we should consider. What I've highlighted in the top green circle is the effects from tidal turbine noise. So what you see is the time to metamorphosis along the bottom axis and the percentage of crabs that have metamorphosed along the y-axis. And the dark line represents where there's noise from a tidal turbine, and the open circles represent where it's silent. So you can really see a real difference there. Again, this was a laboratory experiment, so there's a lot of questions around what happens in an already noisy environment, like a tidal channel. And again, this is crab larvae, so in terms of species specificity, there's a lot of uncertainty, I suppose, around that as well. The final one that I wanted to highlight, I guess, is probably the one that's closest to my heart, and it's where my research background really has come from, is around larval dispersal. So hydrodynamic flow patterns are one of the key determinants of where larvae and zooplankton go and where they can settle or where they find their nursery grounds. So there's been quite a number of studies from a physical oceanographic perspective looking at changes to hydrodynamics resulting from tidal turbines. And I've just illustrated that here on the right-hand side, a piece of work that was done by Matt Easton. And what you see is the Pentland Firth. The island you see is the island of Stroma. So this is essentially where the Maygen development is going to be in the Sound of Stroma. The colours that you're seeing represent the anomaly in the current speed as a result of putting in some tidal turbines. So essentially where you see colours represents either a change positively in increase in flow speed or negatively in flow speed. So the thing that immediately comes to mind to me here is, well, what happens to the zooplankton that you find in the water column? What do these changes in velocity mean to them? And how does it change where they'll be transported further downstream? And building on that, how does that change the biogeography potentially, or the connectivity of that species? So that gets me wondering things like, will we find a greater effect in a channel like this one in the Sound of Stroma, or an estuary, versus an open ocean site? And I guess one of the key points here is that there remains a lot of uncertainty in terms of larval ecology around larval behavior and how that changes from species to species. And so in order to address this specific issue, things like deal vertical migration, selected tidal stream transport, and the swimming abilities of the individual zooplankton might be something to consider. So I suppose those were just a couple of quick ideas to kind of get us started here, and the next speakers will talk in a bit more detail about some of these things. But for me, I really wanted to highlight some of the uncertainties and challenges, and perhaps be a little bit controversial given that this is an expert forum. So the first one for me is the real challenge around scaling up from larval scale, so how a larvae experiences the world on a millimeter scale, all the way up through the dispersal scale, and that's up to tens to hundreds of kilometers, and then translating that into effects on a population. But to me, one of the key things really to consider is how relevant is this issue really? One of the things I've always wanted to do is go and do a back of the envelope calculation on how relevant offshore wind turbines are in the sea, and how much they change larval supply and population connectivity. And I think it's that sort of thing that we need to consider here. How much larvae are we expecting to lose or be affected from tidal turbines or tidal installations versus other factors? And that's things like predation. And just to illustrate that in the bottom, I'm not a lobster ecologist, but I thought it was a good illustration. You've got one lobster can produce thousands to tens of thousands of eggs. So say that equates to 1,000 larvae, and you then get 50 juveniles surviving and one adult surviving from that, and that's in a natural system. So that leads me to question is what's limiting these populations, whether it's larval supply, recruitment, is it a top-down control from something like fisheries, for example? So just in that context, are effects from tidal turbines relevant as to what's limiting the population or not? And perhaps I'll leave that discussion to the end of the forum. So thanks very much.
Thanks so much, Rayanne. That was great. So we're going to hand it over to Brian Sanderson now.
Thank you, Andrea. I'm going to just drill in a little bit on what happens right at the turbine. Okay. First of all, we're going to just think about what's a solid and what's a fluid, and what do these things really mean. Think in terms of continents. We're used to thinking about the land we live on as being a solid thing, but if you were to try and hook a massive crane up to a continent and lift it, it'd be just as pointless as reaching into a bucket of water and trying to lift it and grab hold of the water and lift it out. It was the rock is simply fluid at that large scale, and that is exactly analogous to what we have when it comes to going from the scale that human beings think about when they're playing in the ocean to the scale at which small plankton experience in the ocean. So whereas with the rock, the ratio was the gravitational force to the strength of rock, the equivalent ratio for the water environment in the ocean is the ratio of the inertial forces to the viscous forces. In fact, those two ratios are both Reynolds numbers. So we have Reynolds numbers are large, the velocity and length scale are large compared with the viscosity for something like a turbine or something like a swimmer, an Olympic swimmer. But they're very, very small for plankton at plankton scale. So whereas a turbine or a swimmer essentially experiences the water to the first order of water as though it's an inviscid fluid, is something that is basically infinitely deformable. This is not true at the plankton scale. And one other thing that's very important to realize, the scale of plankton varies by at least 3 orders of magnitude, more perhaps. And so what we mean, so the degree of viscosity is very variable from one plankton to another. Now, if you look at a little plankton, I've got on the right there a picture of the streamlines and little velocity vectors around a copepod. And this one is very reminiscent of a paper that Jeanette Yen and myself and Rudy Strickler published in Limnology and Oceans way back in the 1980s. And we actually calculated strains associated with those zooplankton generated flow fields. Now those strains are much bigger. In fact, that's the strain of the fluid is much bigger that the organism itself creates. It's much bigger than what you'd get in most naturally occurring turbines. And that's something that's important. Next page, please. So I said that to a first order, at the scale of the turbine blade, we actually can describe things, at least into the first order, as though the fluid is inviscid. And I have a plot on the right here where you can see a little magenta line that is a section through a turbine blade, and upstream you have a flow indicated by the red arrows. Downstream, that's background flow again. So sandwiched between I have what actually happens to the current as it flows, as a turbine blade cuts through it. And this can be calculated by, this calculation here is actually made without any friction, without any viscosity. And what we see, and if we were to, we have to introduce something called circulation to remove a singularity at the trailing end of the turbine blade, and that is actually what gives the turbine its lift. It's what makes what lets aeroplanes fly. But we're still, this calculation being envisaged, still leaves a singularity at the front where we get strong velocities. But the thing to notice about this is that most of the problem is described by envisaged no viscosity flow field. And we can see that mostly, apart from that leading edge, the flow field is not really changed much. It's not big strains. And so any little small creature floating through isn't even going to know what's going on. It's inertia. It experiences virtually inertial changes associated with that sort of flow field are irrelevant. The only place where a small creature will even know that something's going on is perhaps at the leading edge of that very sharp blade, because it's a thin foil turbine blade there that I've made. But of course, if you broaden the leading edge, that's less of an issue. But the place where things change, where things become relevant on the plankton scale, is in a thin viscous boundary layer that's right along the edge of that blade initially. Now that is that's a very thin layer. The thickness of the layer generally broadens downstream along the blade, but it broadens as the square root of the distance along the blade. Can separate, you don't necessarily want it to separate. Can generate cavitation, and you don't want that to happen either. Now the important thing to notice about this is that the pressure in that boundary layer is entirely determined by the outside dynamics. In other words, the dynamics of the big scale solution. So the issue here is strain, and the strain is located, and the strain is only very, very close to the blade. And so what that means is that a very, very small number of planktonic, very, very small proportion of planktonic organisms will ever even get into a zone that has high strain. Now remember, many of them are evolved to deal with high strain. Now, cavitation can be an issue. Next slide, please. And that is the one place where pressure changes can be very nasty, and particularly if bubbles are formed and then those bubbles collapse. And on the right there, I've actually calculated what happens with a collapsing bubble. It's a somewhat idealized model, but nonetheless, the blue line shows the normalized pressure, and the magenta line shows the normalized speed. Now, pressure is normalized with respect to ambient pressure. So what this tells us is that the collapsing bubble will create a very high pressure, many times ambient pressure, for a very short amount of time. And this is, in fact, a good way of generating sound. Now, of course, the cavitation risk of a turbine goes down as you put it into deeper water, because it becomes more and more difficult to create the negative pressures that cause cavitation. And any turbine manufacturer worth their salt, if they're building in-stream turbines, would make sure that they didn't create cavitation. Nevertheless, this is one thing that needs to be demonstrated. I think if you're going to put something out, you should demonstrate that you're not making cavitation, and there are two possible ways of doing that. Video footage would demonstrate it. The other way of demonstrating it would be from acoustic monitoring, because turbulence is an incredibly poor generator of sound. It works as a quadrupole generator, whereas a collapsing bubble. It's a dipole generator, and of course the pressures are large, so you'd certainly hear a lot of sound if there were collapsing bubbles. So that is something to bear in mind, a very manageable risk. Next slide. Now, there's a general philosophy that people sometimes throw their hand in the air and say, oh, big is bad, you know, they're putting in monster turbines and therefore this is bad. In actual fact, it depends upon what you mean by big. And the critical thing about the turbine is this: it produces some power P, which will basically equal the pressure acting on the turbine multiplied by the area that the turbine blades sweep through, multiplied by the speed of the blades. So that's a basic relationship. Now, if you look at a hydroelectric dam, what they do is they focus a huge amount of energy onto a very small area blade, so that there's a huge pressure, high speeds of the blade, small area that the blade sweeps through. That definitely is problematic, because as the speeds increase and the pressures increase, cavitation becomes an unavoidable. Shear strain associated with shear at the edge of the blade becomes huge, and so these things are a problem. In-stream turbines are an entirely different kettle of fish. There, for the same area of turbine, it is operating at far lower pressure, far smaller blade speed as a rule. In fact, almost always. In fact, definitely always. So you can actually, if you are running into problems, the solution is always to make the turbine bigger, so it operates. You can get the same amount of power at lower pressure and lower speed. So it's safer to get your power from a big area with a low pressure and a small speed, and this all scales back to the how those viscous boundary layers work on the turbine blade and cavitation effects as well. And I think I'll just leave it at that. I've tried to indicate several things that are important in terms of turbine design, and the key take-home message I leave you with is that it is a very small portion of the area of the water that goes through a turbine that actually interacts with the turbine on the scale of a plankton.
Terrific, Brian. Very, very helpful. Great. Okay, so we're going to turn it over to Brady Quinn now, who's going to really hone in on lobster.
Okay. All right. Hi, everybody. So yes, I'm going to be talking about American lobster, Medea fundi, and specifically considering some of the things that could mediate whether they're at risk due to turbine developments in the Minas Passage or not. Okay, so if you're not familiar with it, this is just a diagram of the lobster's life cycle. So it's split into these two very different phases. The adults that we're kind of used to seeing in the fishery tanks live on the bottom, whereas the larvae live in the plankton. They go through 4 different stages while they're up there, each separated by a molt. And during this time, they can also drift and disperse and maybe connect populations. And a key point is that even though the fishery focuses on the adults, recruitment to fish populations does depend on larval supply. So they can be quite important. And I've sort of tried to narrow down some factors that I think could affect whether larvae, their ecology, could be impacted by tidal turbine development to a few different things listed here, which I'll talk about. And so specifically when and how long larvae are actually present in the water column, their distribution, including how many there are and at what depth they're located, their size, their ability to swim. And I don't have it here, but another thing that will also matter is their dispersal and how that may be affected. And I will touch on that very briefly at the end. So first we need to know when larvae are present. So this is determined by a couple of things. First would be the actual seasonal timing at which eggs that are carried by female lobsters hatch. And so I have here with the red bars an example of the seasonal distribution of hatch for one area. It's actually Advocate, Nova Scotia, which is very close to where the minus channel tidal turbine developments are going on, or planned. And we have here on the x-axis different weeks of the summer, and on the y-axis the proportion of total annual hatch that occurred in those weeks. And this is data from one of my colleagues, Martha Haar, who also works on lobsters. And so what you can see here is that hatch begins near the end of June, peaks around mid-August in this area, or mid to late August, and then ends at the end of September. And other parts of the species range have a similar curve, maybe a slightly different change. So this is the period when you can actually be getting larvae hatching into your area. And then how long they stay there is further influenced by just the time it takes these larvae to complete their development. And this is strongly influenced by temperature. We have a lot of different lab studies that have looked at this and derived some equations that can make predictions of larval development time based on water temperature. And so I've tried to extrapolate here very roughly using some of those results based on, say, mean temperature I found for the entire Bay of Fundy, about 12 degrees. And those are the arrows you see here coming off of the bars. And so I'll just say that a lot of this work is lab-based, but there's been some recent evidence that in nature, in the field, larvae develop more than twice as fast. So I've shown that here as well with the solid arrows. So you might want to pay attention to them. Maybe they're more realistic. And so going from this, then, you could potentially have larvae present and drifting in this area up until, at the latest, early October, I'd imagine. And the majority would be there from start of July until the end of September. And so what that means is that for about 3 months of the year, there's larvae present in the area. This makes up about a quarter of the year. So maybe one way that, so outside of this period, maybe larvae wouldn't really be impacted by tidal power. Within this, if there was risk of or if there was concern of some risk to larvae, developments could maybe avoid the windows here that have the peak hatch. Should we look at? Okay, and the next factor that would be important to assessing potential risks would be the distribution and density of larvae. So the main way this is assessed in studies is with plankton toes. However, most of the data I found for this is for other parts of the species range. I couldn't find anything for the minus basin. What does seem to be the common theme, though, is very low and variable densities. So, for example, I found some number of larvae caught per hour of sampling effort with these plankton toes. And in some cases, you're getting less than one larvae in several hours of sampling. It is very variable, though. Sometimes you'll get into a concentration of larvae. But the overall theme, though, as we heard earlier, one female lobster can produce thousands to tens of thousands of eggs, but we don't see those kinds of numbers of larvae altogether. And so this might mean they do disperse quite widely. Something else as well is that larval mortality in nature is probably quite high. I've seen some estimates based on mass balance models that place larval mortality rates at upwards of 90%, maybe even 99%, and 6 to 30% mortality of larvae per day. And this is thought to be due mainly to predation, stuff eating them. And so this is probably a big factor leading to low densities of larvae. And it might also limit effects of other factors. So if they're this dispersed and so many other things are limiting them, maybe other sources of mortality would be less important if they were a factor. Something else that's also would be important to know, of course, is the vertical distribution of larvae. So what depth are they at? Particularly because tidal turbines could be located at different depths, so on the bottom versus in stream. And the vertical position of larvae does seem to be quite variable. There's reports of them anywhere between the surface and 30 meters or more of depth. And there's different things that influence this. And so one factor would be mixing or stratification of the water column. Larvae have been shown in the lab to not want to cross strong thermal gradients, and there's evidence from plankton cones that they will tend to be found above local thermoclines if they're present, even if these are very deep. It's a little more complicated in more well-mixed areas. Depending on which area or study you look at, larvae are either mainly at the surface in these conditions or they're throughout the water column. There's also differences among different larval stages. So the very earliest stage and the late stage tend to stay near the surface, at least during the day, whereas intermediate stages 2 and 3 don't have as much of a depth preference. They do seem to avoid light in the lab, and when they're found in nature, they're deeper. And so this will result in some differences in where larvae are found over the course of the season. So these kinds of movements would influence whether and for how much time larvae could potentially even encounter turbines. In principle, maybe placement of turbines could avoid depths most frequented by larvae, again, if there's concern. I will note, though, that these observations I'm citing here, some of them come from very basic lab studies. So if you apply this stimulus, larvae moves towards or away from it. And it's not really sure how applicable that is to the things that happen in nature. And it's also what little field validation there is from plankton studies tends to contradict itself among different areas. So there's definitely need for more work on this, and just the actual vertical distribution of larvae in nature remains an open question. Okay, and we also have to think about larval size and swimming. So depending on what stage you're looking at, larvae will usually be about 8 to 15 millimeters in total length, which is definitely still quite small compared to a fish or a human or megafauna. It is somewhat large compared to other zooplankton, though, in the area. So they are quite small, but it's unsure how they could what would happen if they did pass through a turbine. We may hear a bit about some work with this in other plankters and the next talk. But I think it would be good to know for lobster larvae if they can actually go through this, probably with an experiment. Larvae are also surprisingly good at sensing their environment, detecting currents, pressure, temperature, number of factors, and reacting to these. They aren't very strong swimmers in terms of moving forward and backwards, so against currents. But they do react to these, and they do seem to move vertically without a lot of trouble. And it's actually thought that in nature, they might adjust their position in the water column based on the currents they experience, and maybe avoid dangerous conditions, such as currents that would take them offshore, where it's too deep for them to ever settle, and they'd be essentially doomed. And so, given this, there's at least the potential that maybe a larva could detect, say, some of the turbulence or currents from a turbine in its environment, and then move accordingly to avoid it. But we really don't know. Again, cues and swimming abilities are at present. We have limited understanding. So it would be good to confirm this to see if larvae could avoid these things. I would say, based on all of this, though, that the risk to larvae of, say, small-scale tidal testing operations is probably minimal and isn't very likely to have impacts on the populations these larvae support. Now, this doesn't necessarily mean for larger developments there could be potential risks which should be kept in mind. However, again, I don't think we really have the data at hand to assess this risk. So to really say definitively yes or no. So I would recommend some caution, and I do think we need more study just of many different aspects of larval biology. So particularly their density in the regions that are going to be subject to tidal power developments. It would be good to have a baseline, for example, of how many larvae tend to be found in the water column. And then you could compare that later on to after developments from head preceded. It might be one way to potentially get at impacts, if any. And again, just better understanding of how they behave and where they're found, their natural mortality rates, This could give us a better understanding of encounter probability with tidal structures, ability to avoid them, and again, just the relative impact of whatever encounters occur. So just to end, I'll leave you with a couple of implications. So I have collected some data here based on fisheries landings of catch in different years in the region. And what I've got on the graph here, I have different years on the x-axis. And then I calculated the proportion of lobster landings in the entire Bay of Fundy region, which is made up by what's called LFA 35. This is a lobster fishing area, a management unit, and it's the one that contains the Minas Basin. And that's the proportion of all the Bay of Fundy landings made up by that. And then I also figured out what proportion of the Bay of Fundy landings just come from the Minas Basin, and assuming that these represent lobster abundances. And so what you can see, focus on the black line for now, that usually it's about 10% of Bay of Fundy lobsters catches are coming from the Minas Basin. And even within that one LFA, the Minas Basin only seems to represent about a third of its landings. So what this tells us is that then, if there is any risk to developments in the Minas Basin area, it doesn't seem like it should have far-reaching impacts on the whole Bay of Fundy, which is good to know in light of just some controversies that have come up. Of course, though, if there were any impacts within the Minas Basin, it would still be good to avoid these. And then there's also some implications, though, related to larval dispersal. I have some very rough estimates from work that I've done modeling dispersal of lobster larvae in this area. So on this graph here, again, there's years on the x-axis. And then the numbers plotted here would be the proportion of larvae either released from our LFA 35, so the fishing area that contains the Minas Basin, or settling there that end up drifting to or coming from other fisheries, so outside of this area. So for example, the black line would be proportion of larvae released in the Minas Basin that go somewhere else. And the red lines would be larvae that settle in the Minas Basin that come from somewhere else. And so what these results have shown is that this isn't an isolated system. So there actually is quite a bit of potential connectivity of larvae to and from the Minas Basin to other areas in the Bay of Fundy. So for example, LFA 35 supplies on average about 30% of settlers to some other fisheries in the bay. Again, these are primarily self-sustaining, but this 30% could potentially represent something important to them. And of course, this connectivity is going through the Minas Channel, which is where the developments are happening. So it will be good going forward just to keep in mind any potential risks to drift or dispersal of larvae through this might actually have some implications at a wider scale. I will admit, though, that these are preliminary estimates based primarily on physics and more limited biological information. And so I am working during my current degree to improve this system, but it will be worth keeping in mind. And yeah, I'll leave it there. I think I've gone over. I tried to squeeze too much into this, but thank you.
Thank you, Brady. That's great background on lobster. So we'd like to turn over now to Dave Schlesinger to talk about some work they did with zooplankton.
All right, thank you. So this is some work that was done in 2010, 2012, and it was part of a demonstration project where we took a first-generation turbine out into Muskegon Channel in Massachusetts, specifically to look at the effects of turbines on zooplankton injury and mortality. One of the big concerns was being a big fisheries area, whether or not the food chain would collapse as a result of a build-out of mini turbines in the channel, which is bringing water into the major fisheries areas. This is a collaboration between UMass Dartmouth and the Woods Hole Oceanographic. And so, there's been a lot of work on traditional hydroelectric plants, specifically to look at the effects of dams on the mortality and of plankton and fish. For example, the Hoover Dam. Lots of dams in the United States and in Canada. The first thing that came out of reviewing in the literature is it's very, very difficult to make these measurements with large flows of water. And more importantly, it was difficult to determine when animals were killed or damaged, what caused it. The ones that are relevant to tidal turbines would be blade strike, shear stress, and cavitation. Barotrauma, not really a problem because there's no real difference in pressure. The best guess for the mortality range in a really dynamic system like a hydroelectric dam was 5 to 15%, so pretty small. But there hasn't been really any studies on tidal turbines. This is the tidal turbine that we used. It was a first generation free flow turbine, about 1 meter in diameter. This was all done near the surface from a barge about 3 meters below the surface in the summer. Current velocities ranged from up to about 3 knots, so a little bit different than up in Minas Basin, but maybe on the very edges of the velocities that you're seeing up there. We used pretty traditional techniques. We did plankton tows before and after the turbine. We recorded the current velocities and basically worked very, very quickly. We know that the zooplankton survival in a bottle was not fantastic, so we worked very quickly, moving from the channel out back to a lab, and usually they're analyzed within an hour and a half, but we set our limit at 2 hours so that we didn't have a lot of fatalities just due to transport. We did some serial dilutions from the net capture, both before transport and after transport, but working with our colleague at Woods Hole Oceanographic, we were pretty certain that we were not destroying our samples just by travel time. So once we got these samples back into the lab, we were using sort of a one-off system where we were using a flow cell and a microscope to observe the zooplankton in the samples. This was originally developed for the USGS to assess ballast water treatment standards, and it was designed to use to examine very large amounts of water. The samples would pass through a flow cell underneath the microscope, and sequential photographs would be taken to look at the mobility of the zooplankton. And there was also an electrode set up so that we could stimulate them just in case there was some effect of changing temperature or some other effect. So we were pretty confident that we were able to capture differences between those zooplankton that were compromised by passing through the turbine and those that weren't. The nice thing is that most of the software, which turns out to be very important, is open source software and readily available to anyone. So this is some of the results from those experiments. We have particle diameter on the x-axis from 50 to 400 microns. The counts of organisms on the y-axis, and on the second y-axis on the right we have our p-values for both individual size classes and for the entire population that was analyzed. And so we see in the red the pre-turbine counts for each size fraction, and in yellow the post-turbine counts for each size fraction. And the error bars are standard errors, and they all overlap. There's very little difference, no significant difference between pre and post turbine animals. You can see in the yellow dotted line at the bottom the 0.05 0.05 significance level. In green, that's the p-values for the individual size classes, or I'm sorry, in the white, individual p-values based on paired t-tests. So take-home message was that there's really no significant impact of passing through the turbine. What's significant is that the number of particles diminishes, or number of animals, if you will, diminishes greatly as you move up in size class, so that we're only looking at a few individuals once we get to 300 to 350 microns. These are about 5-minute tows, and they were the nets were full of stuff. And you know, it harkens back to the previous speaker, where there's just not that many organisms that are very large, at least not on the lobster larvae scale of millimeters. This was repeated actually about 2 years later with a one-off turbine that spun much faster than the free-flow turbine, on the order of about 200 revolutions a minute as opposed to 60 to 90. And the results were very similar. There was no significant impact from the turbine blades on the zooplankton. I should say that we did notice a lot of megaplankton, like jellyfish. These jellyfish suffered a lot, primarily from blade strikes and shear. I have a video showing the jellyfish really just sliding apart as they move through. But you know, again, that's a size class that's much, much larger than the lobster larvae that it would be important here. So once again, the tidal turbines do not affect the viability of the small zooplankton. And zooplankton risk is very low, we believe. You know, initially this was to look at food web interactions and whether or not a complete build-out would be deleterious to the fisheries in the area, and we think not. We were not able to look at the effects on organisms an order of magnitude larger, on the order of millimeters to centimeters. Overall, we believe that the risk of negative interactions would be higher than for the smaller zooplankton, but still very low, both because of the small density animals and that they don't quite reach that threshold where shear stresses would be important. Along with that, they're very difficult to image. I mean, obviously we could catch them, but with very long plankton toes, which is not practical. The next best thing would be to look at these animals passing through turbines. I don't think the technology is quite at the point where we could do in situ video monitoring. It's close, but not quite there yet. Based on the size of 1 or 2 centimeters, so 20 millimeters, and just sort of a default 1 knot current, we estimated we would need 240 frames per second imaging in order to identify the organisms as they pass through that the plane of the turbine. And that really creates a big problem because that's a lot of images and a lot of data. The big challenge is to be able to have software that can limit the amount of storage needed in the computing requirements by basically discarding all the frames without organisms of interest. And that's a big challenge, but it looks like maybe in the next few years that the technology will catch up so that we can really answer the question. Thank you.
Dave, thank you. That was really, really wonderful. So we're running a little long, but I'm hoping that other people are as sort of wrapped by this as I am. Before we go to some open discussion, and if you do want to type any questions in or comments into the IM box there at the bottom of your screen, go ahead, or we'll just open the microphones. I did want to give Mark Bevelheimer a couple of minutes, and I'm just bringing this on you, Mark. I didn't tell you. We didn't have a speaker specific to fish larvae, but I think that a great deal of what we've heard is obviously very applicable to fish larvae. It's sort of, again, a matter of scale and buoyancy and so on and so forth. So I don't know if Mark or even Garrett would like to say a little bit about their perceptions of fish larvae around tidal turbines.
This is Mark. I don't know if you're referring to the lab studies or some of the some acoustic analysis we did at Verdant.
Any and all, whatever your thoughts are.
Well, the acoustics work was we were really looking at big fish there. So, we have data that could be translated to larval fish probably, but we really didn't know what was there. So there's not much we can say about that. We did do some larval studies about 3 or 4 years ago, and it was just real preliminary things where we had a flowing system in the lab, and we had a couple of blade shapes that were just permanent shapes in the flow channel, and we put larval and young fish by them. They were from 3 to 4 days old up to about a month and a half old, and we were able to do studies and collect larval fish, and we were able to collect the control fish with pretty high survival. So we at least were able to demonstrate that we could do those studies. The velocities that we tested were only a little over a meter a second. So we really needed to bump that up some. But and we did see a little bit of reduced survival in the test fish that were exposed to a blade. But again, the blade was stationary. So those were real preliminary studies. The one thing that I would like to say real quick and that I think is more relevant than those studies, and that's there was a lot of work done in the 80s and the late 70s on compensatory mechanisms related to entrainment of eggs and larval fish in power plant intakes in the Hudson River and throughout the Northeast U.S. primarily. And there's a whole body of literature on compensatory mechanisms and whether or not removal of certain percentages of the egg and larval populations had impacts on the adult populations, eventually the fish communities. And that work was EPRI funded. A lot of it was done at Oak Ridge and other places, and a lot of work was, and it spawned this whole field of individual-based modeling. And so if people want to kind of take that approach and see, understand some of that to kind of ask the big question, does this really make a difference? That's a good place to start. There's still a lot of work going on in that area of individual-based model development and how effects on different life stages transfer to effects on adult populations. So that's all I'll say. Thanks, Andrea, for the chance.
Thank you, Mark. Good points, really good points. This obviously is a big topic we could go on for a long time about. But what I'd like to do is just have people chime in. Questions, comments, etc. If not, I'm going to start, actually. I thought that these were really some good approaches to this. I had a couple of questions or comments. Brian Sanderson, when you talked about, obviously it's to the advantage of the small organisms not to have turbines that cavitate. I just wanted to point out that this is also very important to the developers not to have cavitation on their turbines, because it actually causes damage to the turbine blades. So here's a situation where having what the developers, sort of financial and operational needs comes together with the environmental, protection. So any thoughts on that, Brian?
Oh no, I appreciate that fact. Absolutely.
Great.
I have a question for Dave Schlesinger about the video imaging of zooplankton. And he mentioned that an ideal 240 frame rate per second, which is extremely high for underwater video.
Right.
I just wondered why that many? Is that to get the accuracy so that you can actually see if the blade is hitting it?
Well, I mean, most of the criteria are species based. You got to know what the species is. And so, an organism that's, say, 10 millimeters, you really, in order to identify them, you have to have a very narrow focus. And typically that's on the order of a few centimeters. And if you have something going 100 centimeters a second, just the back of the envelope calculations that we did said, well, there are cameras running around 240 for televisions, for example, and that would allow at least a couple of frames where the organism is in focus and in the field of view. And it's a lot. It is. But for a species identification, we felt like that was sort of the lower limit.
Understandable. Just as you said, very hard to process.
This is Andrea, just following up on that. Yeah, Dave, my mind boggles because we're trying to look at underwater video for fish and marine mammals. And in these highly, energetic and turbulent waters, it's tough enough to do that. So the plankton stuff is mind-boggling. I noticed that you had tapped into some of the ballast water work, looking at trying to look at organisms with large volumes of water. I used to do that. And during that time, there was a big push on some of this image recognition work coming out of a small company in Vermont. I don't know if you're familiar with that. Can't remember the name of the company. But they were doing some great stuff with these high-speed video, very data-rich kinds of environments, but really went a long way towards the zooplankton recognition, image recognition.
Right. I'm vaguely aware of it. I mean, I know they exist. I haven't really looked at it too closely. One approach that we've been looking at is using sort of a modification of particle imaging velocimetry, where we're basically using a sheet of laser light. So we get a monochrome image. We can vary the depth of field by using different line generators, but typically, 1 or 2 centimeters. And it's great because one of the big limitations is being able to get decent imaging in the dark.
Right.
Acoustic imaging is just not fine enough. And as soon as you start adding lights and stuff, you've disturbed the whole system.
Absolutely. If there aren't direct questions for some of the speakers, I'd just like to come back to some of the questions we started off in the beginning, is whether those on the phone have a sense of what we know about the risk to lobster. For example, I was very struck when Brady pointed out that lobster larvae are distributed through the upper 30 meters of the water column. And at least in these deeper tidal channels, on the whole, our tidal turbines are at greater depths. It's probably not true in shallow waters, but certainly the ones we're talking about on this continent tend to be deeper. So that really does speak to a very sharp decrease in risk between the tidal turbines and the lobster larvae. I wondered if people had thoughts broadly, lobster larvae, zooplankton, fish larvae, along those lines. Have we learned anything along the way about what those risks actually are?
I've got a quick question. Oh, go ahead.
Sorry, Anna Redden here. I just wanted, I'm not sure if people realize it or not, but the turbines that are going into the Montes Passage are quite diverse, and some of them are being suspended from flotation units. And so we have a much broader coverage of the water column in that particular test area. In terms of a bigger development, we'll see, who the winners are and what sort of technologies would last. But at least for the test center, a number of different technologies would cover, almost from the top, well, I'd say, 5 meters down to near the bottom.
Really good point. Thank you, Anna. Other thoughts?
I've got perhaps a bit of a question, and maybe Brady can answer it, just about, and it's something that I've always wondered in some of the work that I've done previously as well, in terms of vertical distribution of things like lobster and fish larvae. And I think we've got a really good handle on that in more calm environments than tidal rapids, for example. And I think the reason for that is that we haven't really, nobody wants to go out to, for example, here in Scotland, the Pentland Firth, and sample plankton, because it would be quite an exciting thing to do. It's a harder area to deal with. And I just wonder if in the kind of highly well-mixed turbulent areas, we have a good handle on kind of zooplankton and larval vertical distributions. Because I often wonder if these organisms can swim vertically well enough to stop being mixed all the way through the water column. I don't know what the answer is, but maybe somebody else has got a better idea around that. And that, that might influence, I mean, personally, I don't think that there will be a huge impact on larvae myself, but maybe that might guide our thinking a little bit if we have a bit of clarity around that topic.
It is very exciting. I mean, it's tough because you're right. It's a little sketchy to be out, but I don't think that there's should be a huge risk factor. The question really is what sort of size fraction. What if you're getting the 50 that turn into that one lobster, maybe it is sort of important. My guess is that you would need some sort of direct observations, but actually looking at say diurnal migration up and down through the water column is going to be really tough. I think we need to look at other studies in calmer water, because the turbulence is a lot to deal with as far as mobility of the organisms.
Yeah.
Yeah, I agree, and I guess my question is I guess from ecological curiosity than anything, is if there's studies on diurnal migration or selective tidal stream transport or whatever it might be, if those hold true, where you've got upward and downward velocities in the water column that might be stronger than what these organisms can swim effectively against.
Rayanne, this is Andrea, and this is something I used to do. They are absolutely the mixing, the vertical mixing is far stronger than even the best of these little swimmers can do. Even the larger crustaceans in the water column can't keep up, as it were. So it is most common if you do do vertical plankton tows in these higher energy areas, perhaps not quite as high energy areas as Pentland Firth and Bay of Fundy, but I've certainly done this in places like Admiralty Inlet and Puget Sound, and it's a very uniformly mixed zooplankton through the water column.
Yeah, and I think for lobster larvae too, like I'm trying to think of the studies I've shown I looked at that actually looked at vertical distribution. The biggest one was like way offshore, much different environment than the coastal upwelling ones that we're talking about. I can think of some work, I think, in the Northumberland Strait, and that it's not at the same level as the Minas Channel, but it is a very strongly tidally mixed in the summer. And yeah, I think they were finding lobsters throughout the Cup water column, whereas another area, Magdalen Islands nearby, similar thermal conditions, I guess, but less calmer. They tended to find them more in the surface. Yeah, I don't know that there's a lot more beyond that, but it would be very interesting to know.
Yeah, it's Brian here. From a purely physical standpoint, you wouldn't expect them to be able to. You wouldn't expect them to be able to hold their position in the water column given the vertical mixing.
Another issue is sampling in these high flow areas, and trying to tow quantitatively at different depths at the same time. It would be very difficult and maybe unwise from a safety point of view.
Hey, plankton people are tough. We can take it.
Yeah.
But no, I'm joking. But the one advantage that you have of looking at plankton is it's a whole lot easier to get in, hit a plankton tow, and get out than it is to have big fishing gear over the side. Other thoughts, ideas, overall impressions of where we think we stand on this issue of risk from tidal turbines to these planktonic organisms?
Minimal at the moment, if you ask me.
That's great, Rayanne. I think I heard Dave say the same thing, and I think
I concur
along those lines. And I think Brian has pointed out the scale issues. Any dissenting opinions? Anyone? Even though I think there's still some interesting questions to be answered in the very simplistic, if you will, context of is this a risk that needs to be pursued on a sort of an immediate emerging basis, and should this form sort of a thrust in terms of permitting tidal turbines? Any thoughts there?
This is Mark Bevelheimer. I think one thing, when we think about this, we often think of 1 or 2 or 3 turbines, and we need to remember that these things could be out there in the 20s or 100, and that, when you start doing that, we shouldn't just discount that there's probably not a likely effect, although I generally agree with that. I think we need to remember it could be a larger scale, and that gets back to Rand's talk at the beginning about changing some of the flow patterns, and so you put a lot of turbines in a place and you could really change the environment too. So things to consider. I guess we shouldn't just offhand agree that there's not a likely effect.
That's a great point, Mark. Thanks for underscoring that. And yeah, is there a fairly general consensus that if you were to go from sort of 1 turbine to, let's say, 100 as a huge build out, you and you considered there was some very small risk per turbine, you could probably just multiply that. It still stays a very small number, but that the issue of change of flow becomes the overriding, and therefore distribution of plankton becomes the overriding potential issue. Does everybody pretty much believe that?
I would accept that, but also say that this is actually what, if you're changing the current patterns, that's a change as opposed to a risk. A change. Changes in the environment happen in all sorts of ways for all sorts of reasons, and sometimes there may be winners and losers depending on how you look at it. So I see that as a change as opposed to a risk.
A great point, Brian. Thank you. This, we don't know. Tidal turbines may actually make things better for certain plankton. Certainly, if you're thinking of areas that are somewhat limited with vertical mixing for phytoplankton, maybe there's actually advantages. Really good point to bear in mind: change versus effect.
Can I just chip in something, because I agree quite a lot with what Brian's just said around changes and the scale of change. The other thing to remember is that a lot of plankton are meso-zooplankton, so their life cycle is such that they're not in the water column the whole time. And at least here, when you assess any impacts, you're looking at impacts on a population very much. And so in order to think about that, there's a lot of it's a slightly more complex issue, I think, because you've got a two-phase life cycle. And so there's a whole, all of the other things potentially affecting the adults of these species, as opposed to the zooplankton and larvae themselves. And that's why I think it's really important to have an understanding of what the limitations on the populations are in the first place, as opposed to specifically will tidal turbines impact zooplankton. I think those are maybe the kind of whole population, whole life cycle things are perhaps, in terms of consenting, potentially the more challenging ones. Whereas I think around zooplankton, it's really, there's a lot of interesting science questions there, but I don't know that it's at the moment, of immediate relevance to the consenting process.
Brady here, just kind of a related thing to looking at the whole life cycle and thinking about larger scale developments. I think one thing we want to consider, that ought to be considered too, is the settlement stage of a lot of these, the zooplankton that we know actually settle out to be benthic adults. I'm thinking of lobster. We generally think that the juvenile lobsters, they want cobble bottoms, like rocks and places to hide from predators or each other, and that tends to be limited. And I kind of wonder, I don't have a good sense of this, but in, say, an area like the Minas Basin where you get a lot of sediment just in the water and really poor visibility, I wonder what the risk would be to a large scale, hundreds of turbines, for example, at the channel. How that could change sedimentation and, as a result, available bottom habitat, for example. So you could actually get to, I don't know what the risk of this is, but so say there's not as much place for lobsters to settle once they're done eating plankton, and what kind of effects could that have?
A great point, Brady. And putting that in sort of a larger context, we tend to talk about tidal turbines and that idea of sort of energy removal and changing flow in an area. Happily, this is one of the issues around tidal turbines that it lends itself quite well to numerical modeling. And there are quite a number of modeling efforts that have gone on, and they're getting more sophisticated all the time, of putting multiple turbines in an area and looking at things like changes in flow and changes in sediment transport. So that is definitely one that can be addressed through the modeling process. What we see so far, and obviously it's water body specific, but you have to get to a lot of turbines before you start to see any effect one would ever measure. So it's a really great one to keep in mind. that early small numbers of turbines are highly unlikely to have these kinds of effects. But if you ever did want to go to very, very large numbers, it needs to be looked at, for sure.
Anna Redden here. Just want to comment that a couple of papers have recently come out about that for the Bay of Fundy, and it would take like thousands of turbines to really have much of an impact on that from the work that I have seen. So I agree with Andrea that it's probably not something that we need to be worried about at this early testing phase.
That's right on, and that's about what we've seen in Puget Sound. It runs to the thousands, exactly, which no one's going to ever let you permit for different reasons, I don't think. Okay, other thoughts? Otherwise, I think we're going to start to wrap this up because we want to be mindful of people's time. Do you want to go to that next slide, please, Amy? Thank you. So, yeah, just a few minutes if anyone wants to chime in. I do want to tell you, and I should have said this at the beginning, but this expert forum has been recorded. We will check with each of the speakers and to ensure that you're okay with having your slides put up on Tethys. And the slides and the recording, we may edit it a little just to take out some of the transitions and make it a little tighter, but otherwise that'll go up as is at that URL that you see up on your screen. And I know there's a few of you that haven't been very engaged with some of these processes, and I really would encourage you to go to tethys.pnnl.gov, and you will find there's a tremendous amount of literature associated with environmental effects of marine renewable energy devices. So, any other thoughts anyone has?
I just wanted to just say thanks to Andrea and Amy and Jonathan, Nikki, and others who have helped make this happen. I think this has been a really good forum for discussing this issue. It's really helpful, and it'll be great to have it online.
Great. Well, Anna, thank you for lighting this fire. I'd also like to point out that once these slides and audio are up on the page, that page of Tethys, like all others, has a commenting box. And if you have thoughts after the fact or want to direct others with interest in this topic, please go to that commenting box and type in a comment. And particularly if we think one of the speakers or other participants on this forum can be helpful in moving that discussion forward, we get notified of those comments and we'll pass them on and let them know that they're there. So I think it's an opportunity to sort of keep a discussion and a forum going online. Okay, well, we've got an hour and a half, slightly longer than planned, but if no one has any other thoughts, I'd just like to thank everybody for taking the time, especially thank our speakers for some thoughtful, approach to this, the slides, the presentation. And with that, I guess we'll conclude the forum.